Hello, my name is Weijian Yang, an assistant professor at Electrical and Computer Engineering at UC Davis. I work in the interdisciplinary area across electrical engineering and neuroscience. Understanding how our brain works is perhaps one of the biggest challenges in science. One of the bottlenecks I see in brain research is the lack of advanced tools to characterize and to modulate the brain. Standing at the scientific interface between engineering and neuroscience, I'm devoted to tackle this challenge, to develop engineering tools, and to decode how the brain works. We develop optical methods at multiple spatial scales, from benchtop microscopes such as high-throughput 3D two-photon imaging systems, to wearable optics such as miniaturized microscopes that can be worn by the animals, and to implantable devices which can sense the neuronal signals with minimum invasiveness. Using these tools, we study how animal behaviors emerge from neural activity. We have demonstrated new approaches to implant new memory into the brain, to trigger memory retrieval, and to modulate animal behaviors. As an important step towards deciphering how the brain works, our work could lead to effective solutions to treat brain diseases and to enhance human learning capability. Finally, using what we learn from neuroscience, we aim to make the next wave of innovations in artificial intelligence. This is an exciting era where we have powerful tools to innovate at the frontiers in both engineering and science. We are excited to see the next new inventions in engineering and new discoveries in science.
Hi, my name is Josh Hyatt, and I'm an associate professor here in electrical and computer engineering at UC Davis. My work is in the area of nanoscale and molecular electronics, and my group really focuses on three sub-areas within this field. The first is on developing a fundamental understanding of the charge transport and energy conversion properties of single molecule systems. So to do that, we focused a lot on instrumentation development and new characterization techniques. As an example, we've recently developed a system that allows us to integrate single molecule Raman spectroscopy with single molecule electronic characterization. That means that we can get information out of both the electronic states of a molecule and the chemical properties of a molecule simultaneously. So this is going to allow us to look at things like how small changes in molecular configuration affect the quantum transport properties of the system. The second area that we work in focuses on using our ability to make contact to and electrically interrogate a single molecule to try and create biological diagnostic and sensing systems. For example, we recently took a single DNA probe and used it to look at RNA sequences from 4 different types of E. coli. And each one of those changes caused a significant change in the electrical properties of the DNA RNA duplex that we measured. So this is useful for creating systems that will allow us to do pathogen detection and identification in real time. The third area that we work in is trying to scale these systems up. One of the most recent areas that we've expanded into is trying to use DNA origami as a platform for constructing integrated electronic circuits and devices. And so we're using that to make things like DNA-based nanowires to try and control the conductivity of them and to create memory arrays based on DNA origami. Thank you very much.
I'm Erkin Şeker. I'm an associate professor and graduate program chair of electrical and computer engineering. I lead the multifunctional nanoporous metals research group, where we merge nanotechnology and microfabrication. In our research, we primarily use nanoporous gold coatings that are obtained by an atomic self-arrangement process. This is a relatively new material and has mostly attracted attention for catalyst applications. Surprisingly, the biomedical potential of this material was largely untapped, and during the recent years, our group has become a leader in this field. One of our research efforts is on the development of neural interfaces. These devices are used for monitoring and modulating neural activity that dictate many physiological and cognitive processes. Here, the nanoporous gold electrodes offer novel functionalities such as electrically triggered release of pharmaceuticals, for example, for managing epileptic seizures. Another research effort in our group is on engineering electrochemical biosensors. In these sensors, capture of nucleic acids from pathogens or as a disease marker generates an electrical detection signal that can be easily interfaced with handheld instrumentation. During the last few years, we have been incorporating these technologies into tissue chips. For example, we study neuroinflammation that is common in many nervous system disorders and injuries. We also work on developing a miniatures model of the gut-brain axis for studying the influence of different diets on neural activity that influence high-level functions like mental states. Taken together, I expect our multidisciplinary research to have a broad impact on the development of porous materials with tunable properties for high-capacity batteries, multifunctional electrodes for the study and management of neurological disorders such as epilepsy, electrochemical biosensors for point-of-care diagnostics, and tissue chips to identify mechanisms and therapeutics for disorders such as epilepsy and Crohn's disease.